Okay, guys, just getting ready to start here with unit, or excuse me, our last lecture of Unit 6. This lecture will be on the blood vessels. So let's dive in. This is our last lecture of and discussion on the cardiovascular system, where we're going to kind of finally put things together. Thinking back to our last few lectures, we've talked about the blood, one of the three components of our cardiovascular system. Last lecture was on the heart, okay, which pumps that blood, and where that blood goes from the heart is into our blood vessels. So we've talked a little bit about blood vessels, especially things that are the blood vessels that are closely associated with the heart. And the whole function, as we've talked about since the beginning of this unit, of major function anyway of the cardiovascular system, is to allow for exchanges of nutrients and waste. Okay? And that's because, remember, cells of the body, most anyway, cannot move around and get their own things, cannot get their oxygen, cannot get their glucose, cannot drop off their waste to the lungs to be exhaled off for CO2 or things like urea and creatinine that need to be peed out. You can't just deliver them to the kidneys. So the blood vessels are going to be the conduits to provide that action, okay, or those actions, I should say. So as it says there, blood vessels contribute to homeostasis by providing for the flow of blood to and from the heart and exchange of nutrients and waste in tissues. Blood vessels also play a role in adjusting the velocity and the volume of blood flow. So when I kind of break down that sentence, velocity refers to how fast blood flows in this case through structures, something that does have a very important role in our body. And then volume refers to the amount of blood, which if you think back to when we talked about blood, we learned that about half of our blood is just strictly water. By changing water balance and water levels in our blood, we can change the amount of blood that we have. Now, three major types of blood vessels exist. We've kind of talked a little bit about arteries and veins. We learned in our last lecture on the heart that arteries, think away, we call these efferent vessels, meaning that they carry blood away from the heart. And we talked about the fact that typically that blood is going to be oxygenated. And I say typically because, as we know, the pulmonary arteries do not. The pulmonary trunk, pulmonary arteries carry deoxygenated blood, just like the pulmonary veins do the opposite of what normal veins do, in that they carry blood that is oxygenated back to the heart. And typically, as we know, veins are going to carry deoxygenated blood. And we describe veins as the afferent vessels, whereas the arteries were the efferent vessels. In between arteries and veins are where we have what we call capillaries. And I always say this is where the magic happens. These are microscopic, thin-walled vessels that connect arteries and veins. But more importantly, this is the reason I say that they're the site of the magic, is because they're the location of exchange. And now, at the bottom here, we have an illustration on the left of an artery. And we typically use red to denote oxygenated blood, vessels that carry oxygenated blood. So that's why this artery is colored red. Over on the right, there's a vein. Typically, we see veins colored blue because they typically carry deoxygenated blood. And on the bottom there is our capillary. Okay. Now, there are a lot of similarities between these three vessels, but more so similarities between arteries and veins. Okay. And that's why it says here, arteries and veins composed of three layers, or as you'll see sometimes the term tunics is just the same thing as a layer. Okay. Names of them pretty straightforward: interna, media, externa. If you just add the letter L at the end of those, it tells you exactly where they're located. Interna, internal; media, medial in the middle; external or externa on the outside. Okay. Now, with the interna, okay, it's not a singular layer. It's actually multiple layered, as you can see here with these three sublayers. Now, we also see that arteries have a layer called the internal elastic lamina that veins do not have. Okay, so let's just read through them. Okay, the tunica interna, also you'll see it sometimes listed as an intima, forms the inner lining of a blood vessel and is in direct contact with blood as it flows through the lumen. Its inner layer is composed of simple squamous epithelium that we call endothelium, and this is just continuous in the exact same setup as what we had in our heart, which we learned the other day is called the endocardium. Surrounding it is a basement membrane of loose connective tissue separating the outer layer, known as the internal elastic lamina. As I mentioned, the internal elastic lamina we see in arteries, we don't see it in veins. And the function of this layer, it looks like Swiss cheese, and it's made up of thin sheet of elastic fibers. The holes allow for diffusion of materials. Okay, think of it like, to me, it looks like, a dish strainer. When you make macaroni and cheese or spaghetti, boil the noodles, you dump them into the strainer or the cone or whatever you want to call it. Water's going to flow through easily. The noodles won't, and stuff can easily pass through these holes that isn't too big. Okay. Now, down at the bottom, our capillary, we see it contains that endothelium, very thin layer of flat-shaped squamous cells, and then a basement membrane. And that's all that makes up the wall of a capillary. And as I said, they're very thin-walled. The thinner they are, the more easily stuff is going to be able to pass through. Okay, for example, oxygen that we bring into our body must leave the bloodstream and go into our tissues, so it has to leave the blood traveling through the walls and into the interstitial space. Well, if it had to pass through all these layers of an artery or the layers of a vein, it'd be a lot more difficult than it would with our capillaries. And so that's the structure of a capillary. Pretty simple structures compared to our arteries and veins. Okay. Now, something I always point out, you notice over here on the right, we see something in a vein that we don't see in arteries. Folds of the tunica intima, specifically the endothelium, create valves. Valves are important for what we call venous return. Allows blood to flow up or through them, but not backwards, kind of like the valves in the heart. They prevent what we call retrograde or backwards flow. And we don't see valves in every single vein, but a good number of them do have those veins. So that's one difference between an artery and a vein. I'll get to a couple more here in just a second. Tunica media is a middle layer. It's usually the thickest. It's made primarily of smooth muscle, and the function or principal function of it is to mediate what we call vascular tone by performing what's called vasoconstriction, vasodilation. Vasoconstriction is where the smooth muscle contracts and basically kind of pinches, like kicking a garden hose, reducing blood flow through the lumen, because the lumen, which is the space internal where the blood flows, it gets smaller, so less blood can flow through it. Okay. Vasodilation is just the opposite of that. When those muscle cells relax, the lumen gets bigger and more blood can flow through. Now, the terms vasoconstriction, vasodilation typically are reserved for action in an artery. There is what's called venoconstriction, venodilation, which takes place in veins. Okay? If you haven't already kind of figured it out, looking at the amount of smooth muscle in arteries versus veins is another big difference between arteries and veins. Arteries have a lot more and thus have a better ability to contract or constrict and dilate than veins, which are much more wide open, and that's because they have a lot less smooth muscle. And there is another elastic lamina. This is called the external elastic lamina, which is another layer of Swiss cheese that we find in arteries that we don't find in veins. Another difference, and that's found between the smooth muscle and finally the tunica externa. Tunica externa, our outermost layer, consists of elastic and collagen fibers. Elastic fibers allow it to stretch. Collagen fibers help hold everything together. In your larger arteries and veins, they're actually going to have blood vessels in this layer. So that's kind of crazy. Got vessels that have vessels. We call these vasa vasorum, or vessels to the vessels. Okay, kind of think of them like the coronary arteries and veins that we found in the layer and surrounding the heart, providing nutrients and moving waste from the heart. And that's because just like what's in our heart, that blood that's inside the lumen isn't going to be staying stagnant long enough to allow stuff to diffuse out and into those outer layers and outer cells. And function of the externa is to help anchor vessels to surrounding tissue. So those are the structure of the arteries and veins, and capillaries at the bottom. You hadn't, I kind of mentioned it, but I didn't really point it out too much. Another big difference between arteries and veins is the size of the lumen is much wider, much bigger in veins than it is in arteries, and all that has to do with the thickness of those three layers. We see arteries have a much thicker wall than veins, and that actually allows veins to not necessarily store blood, but kind of accommodate it. I believe it's about 60% of our blood, just like when we're hanging out, resting, watching this video, sitting around doing homework, or just, watching TV, whatever. About 60% of our blood is, found within our veins. But as we do things like exercise and, need to route blood flow to things like our skeletal muscles, the ability to venoconstrict decreases the lumen, which then helps, change that percentage down so we can get more blood to places that need it more than our veins. Okay, what we're going to do here for the next couple slides is talk about each of these types of blood vessels a little bit in greater detail and learn a little bit more about the different types, starting with arteries. As previously mentioned, they are the efferent vessels that carry blood away from the heart. Structurally, walls of an artery will have three layers of typical blood vessel. It has a much thicker muscular to elastic tunica media than we see in the other vessels. Due to their plentiful elastic fibers, arteries normally have high compliance, which means that they have the ability to stretch easily, expand without tearing in response to small increases in pressure. That's especially the case for the first of our three types of arteries. The first type, called elastic arteries, which we've seen the term elastic a lot this semester. When we always think the ability to stretch, but also to recoil. These are the blood vessels that we talked about, like the aorta the other day. As far as arteries go, they're the largest. They're the ones that are closest in proximity to the heart. They have that ability to stretch without injury, and that's going to accommodate the surge of a lot of blood forced into them when our heart contracts. But then they also can recoil. And that's because they have a lot of elastic tissue, much more so than they actually have muscle. This includes the aorta I mentioned, brachiocephalic is in our upper chest, common carotids, which are in your neck, subclavian, which is in your upper arm-ish area, upper chest, vertebral runs up through our neck, pulmonary, that's the one that we talked about, carries deoxygenated blood to our heart, or excuse me, to our lungs, the common iliacs, which distribute the blood to our legs. So these are kind of the biggest, think of them, great size, greater than 3/4 of an inch. That's very large. A cool thing that they do when our heart is relaxed, the walls of these elastic arteries helps to force blood in a forward direction. And that's because they can snap back and push that blood forward as they, snap back due to that elastic fibering they have. They also smooth, jerky discharge of blood from the heart, help maintain pressure, and cause back pressure that closes those semilunar valves. Due to their elastic fibers, they can stretch when blood is ejected from the heart. They momentarily store mechanical energy, allowing them to function as what we call a pressure reservoir. Elastic fibers then recoil and convert stored energy into kinetic energy as blood is pushed forward. So basically that just allows for blood to continuously be circulating through our arterial circulation even when the heart is not contracting. From our elastic arteries, blood will travel into your muscular arteries. These are medium sized, so less than 3/4 of an inch. They're called muscular arteries because they've got a lot of muscle, 40 layers or so, which constitutes about 3/4 of the wall's thickness. They've got a lot less elastic tissue than what we saw in the elastic arteries. And the function of them is to distribute blood flow to the parts of the body that need it by performing vasodilation and vasoconstriction. They help propel blood onward during ventricular diastole, which is when the ventricles are relaxed. They contract when stimulated by the sympathetic nervous system, help stop bleeding, raise blood pressure, and shunt blood flow. As far as what we'll see in this lecture, the vessels in your arms, like the arteries in your arms and legs, most of the arteries that have a name, brachial artery, radial ulnar arteries, femoral arteries, those are muscular. And arterioles are where blood goes next from those muscular arteries. These are the smallest arteries. Tunica media is a much thicker in proportion to the lumen than that of your larger arteries. Allows them to function as resistance vessels. That's kind of their nickname. They have high degrees of ability to constrict and dilate, which helps regulate blood flow into capillary beds and help regulate blood pressure. Terminal end, so the very end of an artery, this can be arterial, it's called a metarterial, and it tapers, which means it kind of gets skinnier towards the junction with the capillary. Most of our body receives blood flow from more than one artery, and in some cases we see two or more arteries uniting, to supply the same body region. We call these an anastomosis. The plural form of that word is called anastomoses. We actually talked about these in our heart lecture with the coronary arteries. And the function of these is to provide alternative routes for blood to reach a tissue and organ. We see a lot of anastomoses in veins and venules as well. From arterioles, blood is then going to travel into the capillaries, which once again is where the magic happens, transfer of nutrients and other vital substances between our blood and cells. Term capillary actually means hair-like, and we can describe them as connecting the arterial and venous sides. We talked about the fact that they have really thin walls to aid in diffusion and exchange. Most only contain that intima, single layer of endothelium with the basement membrane on the outside. Of the 50,000 or so miles of vessels in our body, capillaries make up the largest and the predominant amount. And diameter of a capillary's lumen is just larger than one red blood cell, allowing for rapid gas exchange. We can see that here in our pictures at the bottom. We see in this case a couple red blood cells. Here's one, about the same width, just a little bit wider, okay, than at least one of them. Okay. Metabolically active cells like your brain, heart, and kidneys are going to have a lot of capillaries. Those that are metabolically inactive, like bones, tendons, and joints, have very few. And then places like our lens and our cornea and our eyes have none. Precapillary sphincters are rings of smooth muscle that regulate blood flow through a capillary. So they can either basically pinch the capillary shut or relax and allow blood to come into a capillary. And we find them at the arterial-capillary junction. Okay. There are three types of capillaries. The most common type, most of our capillaries are this first type, continuous capillaries, where they have the cells that form the wall. I'll zoom in. These endothelial cells are held together by way of tight junctions, which makes them fluid tight. Okay, I talked about this back in our lecture on the lymphatic system, how the cells come together and butt end to end. Okay, whereas we talked about the capillaries of the lymphatic system, how they overlap. Okay, well, these don't overlap. They're going to be basically just butted, next to one another, and they create an uninterrupted tube. Now, there are very, very, very tiny little kind of slits or clefts. You can see these little passageways. Okay, we call these intercellular clefts. Okay, inter meaning between. So these are the clefts between the cells, and I'm talking itty bitty. Okay, this does allow small things like glucose to pass through. Okay, like I said, these are the most common type. We have them in our brain, our lungs, skeletal and smooth muscles, for examples. Okay. Next type are called fenestrated. They have continuous walls between the endothelial cells. So they're like these in that they are going to be butted up once or next to one another. But what's different is the cells, and you can see them, have tiny little holes, or what we call fenestrations, or you can call them pores, okay, that make them leaky, okay. This increases their permeability, which means they allow certain things through more easily, to allow for high transfer rate of substances into or out of a capillary. We have these abundantly in our kidneys, which is where we filter our blood and make urine. We pull water and other stuff from the blood. And by being able to pass through these holes, it allows for a lot to occur at the same time. Yeah, buddy. Hang on one second. All right, sorry about that. I'm trying to hide in the storeroom and it didn't even work. Still found me. Okay, so fenestrated capillaries. Place like the kidneys allow a lot of transfer of nutrients, or waste out of the blood, form into urine. And then the villi of our small intestines, where we also have them, which is where we do a lot of absorbing, bringing stuff that we ingest into our body, things like water and glucose, whatever it is that we're eating and drinking. Final type, you can see, very holy. We call these sinusoids. They have really big holes, or sinuses, I guess is what they call. Just so big that even platelets big proteins, even blood cells can pass through these holes. We find these in only special places, our liver, which is where we make our, things like albumin, fibrinogen, our plasma proteins. The bone marrow, which we learned is where we make blood cells, and the spleen, which is where we, can remove blood cells from the circulation as, like red blood cells break down. And so this is where things can, that are big, like our cells and proteins, can leave or enter the blood for the first time if they're coming in or leaving blood, possibly for, ever. Okay. So not very common, but super, super important in their own right. Okay. From capillaries, blood is then going to travel into our veins, the afferent blood vessels that deliver the blood back to the heart. Now, there are several types and features associated with veins that exist, but it's not quite as rigid and straightforward as what we see with arteries and capillaries. Our smallest veins are called venules, and there are two types that exist. Post-capillary, they're the ones that initially receive the blood from capillaries, which makes sense. Post means after, so these are venules that receive the blood after it runs through the capillaries. They measure about 10 to 50 microns in diameter and have loosely organized intercellular junctions, and that makes them very porous, which actually allows for them to do exchanging of nutrients and wastes. They also allow white blood cell emigration, which is where white blood cells leave the blood so that they can go, fight infections and stuff like that that are, taking place outside the blood. From our larger, our next type of venules are called muscular venules. They're in size 50 to 200 microns in diameter, have thicker walls than post-capillary venules, and that's because they've got all three layers. They are still porous enough to allow white blood cells to pass through them. Thin walls of both post-capillary and muscular venules allows for them to easily expand and serve as excellent reservoirs for blood accumulation. While veins do show structural changes as they increase from in size from small to medium to large, structural changes are not as distinct as they are in arteries. So we don't have those different categories other than the venules. In general, veins have very thin walls relative to their total diameter, range in size from 0.5 millimeter in our small veins to 3 centimeters, which is actually over an inch in our largest veins. Many veins, we talked about this on our first slide, especially those on our limbs, have valves to help prevent gravity from pulling blood back toward our feet, formed from folds of the tunica interna. In our last lecture, we talked about the coronary sinus. It's an example of what's called a vascular sinus or venous sinus, which is a vein with a very thin endothelial wall. It has no smooth muscle to alter its diameter. And so in a vascular sinus, the surrounding dense connective tissue replaces the tunica media and externa in providing support. Other examples would be the dural venous sinuses, which are up in our brain, carry deoxygenated blood back down from our brain towards the heart. We've got more veins than we have arteries. That's because we see a lot of them in pairs, accompanying our medium to small size muscular arteries. These double sets of veins escort the arteries and connect with one another via venous channels called anastomotic veins. Our subcutaneous layer deep to the skin is another source of veins. We call these superficial veins. They're the ones you can see through your skin. They course through the subcutaneous layer unaccompanied by parallel arteries. And we have a lot of connections or anastomoses that exist between those and the deep veins to travel between our skeletal muscles. So there's a connection that exists between superficial and deep veins. Down at the picture, I didn't, obviously put pictures of different sized ones. I found these online showing you the valves, which is kind of cool. These are actual cadaver veins that have been cut two different ways. This is a transverse section, so we're looking down into the inside of the lumen. We can see the valve flaps there. This is a longitudinal cut, so it's cut down the length of it. We can see the same thing, just at a different angle to show those things off. All right, now before we dive in and look at the different types of blood or the different types of arteries and veins, better kind of talk one last slide here to get us ready for that point. And on this page, we'll talk about what we call circulatory routes. So, reading through, arteries, capillaries, and veins are organized into circulatory routes that deliver blood throughout the body. Two major circulatory routes are the systemic and pulmonary circulation routes. We've talked about these actually back in our last lecture. We learned that the left ventricle and the left side of the heart was associated with the systemic circulation, where we pump blood out into the aorta, and then that blood is going to be distributed to the body to deliver that oxygen, to deliver those nutrients, and then that blood, after performing gas exchange, returns back to the right atrium. We also talked about the right side of the heart and how it is the circuit for pumping blood to our to our lungs, that blood being deoxygenated, and we then pick up oxygen, dump off the CO2. So that's the pulmonary circulation or circulatory routes. And then that blood, once it's become oxygenated, returns back to the left atrium. Those are both illustrated here in our picture on the bottom left, pulmonary shown in blue on both the left and right sides of our page. And then down here at the bottom, the systemic is all of this in yellow, delivering blood to organs like our liver and our kidneys and skeletal muscles and so on and so on. And now, we talked about this thinking back to our lecture on the endocrine system when we first discussed what we call portal system. Typically, blood flows a pretty straightforward, simple route. It leaves the heart, we then pump that blood into arteries. Blood then travels out into our capillaries for exchange, then is picked up by the venous circulation and returns back to the heart. Okay, that's the simplest, easiest route that blood can flow back, or away from and back to the heart. And there are some variations that exist, one of which is shown right here with this, what we call hepatic portal vein. It allows blood to flow out into arteries and then into a capillary bed. But instead of going into normal veins, there's at least one in our brain between the anterior pituitary and hypothalamus. There were two, what we call portal veins. And portal veins connect one capillary network to another capillary network. And the function of the hepatic portal vein is to allow for nutrients that we absorb, like in our small intestine, jump in the blood and then get carried directly to our liver, because our liver is where we metabolize and we process those things. Once the blood flows through that second capillary network, it then is going to be carried back to the heart by regular vein situation. And so what's pictured here is the hepatic portal system. What we talked about previously was that hypophyseal portal system. On the right is an illustration of what's called an arteriovenous anastomose, a thoroughfare channel or a shunt, all the same thing, which occurs when blood flows directly from an arterial, skips through the capillary network and travels into a vein. Okay? And that's because those precapillary sphincters, those rings of smooth muscle that are located between an artery, or excuse me, the arterial side and the venual side, they contract, and therefore blood will not be able to flow into the capillaries, so it skips through. Okay? This type of route occurs in our fingers, toes, and ears when we get cold, and that helps reduce heat loss, because the closer blood gets to our skin in places like our fingers, toes, and ears, the more likely we can lose heat to the environment, and that makes our whole body, temperature drop. We don't want that, so we sacrifice things like our fingers, toes, and our ears, which unfortunately, if we are, exposed to cold for too long, can become frostbitten. Okay, at this point, what we would do if we were still meeting together is go through, and as you can see, I've got pictures with things that we would color. And this actually takes quite a few days, about two and a half class periods really to get through it all. So it's kind of slow moving. It would take me a couple hours probably to do it. If I was in my house, probably not, since I don't have to wait on you guys. But I've already colored them. And so if you go to, let's see, your unit 6, you come down to where it says vessels coloring, click on that link. I've already colored them and posted the pictures. So what I would recommend is you guys follow along and color them yourself as well. Okay. I'm not going to go through and color them in the lecture because, you know, it's pretty easy to do it on your own, and it won't take you very long to just, you know, do the exact same colors. What you'll notice is for a lot of pictures, I leave stuff completely blank. And those are vessels that you're not going to have to worry about knowing for the exam. And just color what's colored and what's listed in the side of the page or at the top of the page. And what you will not have to worry about knowing for the exam are things that are listed. You know, for example, I'm not going to ask you to know that the celiac trunk feeds the liver, gallbladder, stomach, spleen, pancreas. Main thing is looking at the pictures and identifying the arteries and then eventually the veins. So what you can do is go through and color them all. Pull my screen back up. All right, now there is one exception which you won't find in that link, and that is this slide. And I just haven't found a resource. I don't like the coloring picture from what I use, the book that I use for all of these, that I can use for the arms. And the arms and the legs, as far as veins go, there's a lot, and especially the arms. There's a lot in a small amount of space. And so we've got here a picture, superficial and deep view, or excuse me, deep artery, or excuse me, veins. Wow, veins in the anterior view. Okay. I'm not going to worry about you knowing this set of pictures for the exam. Used to doing it, was just a kind of disaster. Look, it's so hard to tell them apart. I just wish I could find a better picture to color so that it would make a little bit more sense and it'd be a little simpler, but I just haven't found one yet. So you could look at it. It might be something you want to use someday in the future, but for now, don't worry about knowing it. Okay. Just study all the rest with all those pictures that I've already colored. Okay. Now here is a kind of special one, the portal system. You want to kind of read up on this one a little bit more. Pay attention to the way that blood flows. I've included kind of a schematic on the direction which blood flows as it all moves towards the portal vein, which then trains the blood into the liver, and that delivers the nutrients and stuff that we've absorbed from places like our stomach, our large intestine, small intestine, before that blood has to flow back to the heart. Once that blood flows through the liver, it leaves the liver and dumps into the inferior vena cava on its way into our right atrium. Okay. Very last slide we'll talk about, this isn't in that, and it's not a coloring slide, but it kind of helps summarize something we talked a little bit about in our last lecture, and that's how our circulatory cardiovascular system has to change after we're born because of things that change about us. And so let's just read through and it'll make more sense as I roll through. The circulatory system of fetus, what we call the fetal circulation, exists only in the fetus. It contains special structures that allows the developing fetus to exchange materials with its mother. This system, the fetal circulation, differs from postnatal or afterbirth circulation because our lungs, kidneys, our GI organs, so stomach and intestines do not begin to function until birth. Remember, we're not breathing, we're not eating, we're not doing those things until we get outside of mom. Okay? And that's because we don't need to. Our fetus, or us as a fetus, we contain or obtain O2 and nutrients and remove CO2 and other waste into maternal blood because of the placenta that exists inside of the womb along with us. And placenta is a sac that develops inside the mother's uterus, attaches to a baby's belly button, what we call the umbilicus or navel. Now the belly button itself is the remnant or scar that's left over once that umbilical cord has been cut. And so communication with the mother's cardiovascular system occurs by way of small blood vessels emerging from the uterine wall. Umbilical cord contains blood vessels that branch in the placenta. Waste from the fetus diffuse out of the capillaries into spaces containing maternal blood. in the placenta and then into mom's uterine veins. Nutrients are going to travel the opposite direction. So it's kind of funky. We got veins carrying nutrients with babies, and so it's kind of flipped here. Okay. Blood will pass from a fetus to the placenta via two umbilical arteries. These are branches of the internal iliac arteries located within the umbilical cord, which we can see. We've got two pictures. This is circulation at birth. Here's the belly button, the umbilicus. Okay, here's the belly button, but notice there's something attached to it. There's the cord. Here's the placenta inside of mom. Okay. Here are those two umbilical arteries. Notice the direction in which the arrows are going towards and into the umbilical cord. Okay. So blood passes from the fetus to the placenta via two umbilical arteries. These are branches of the internal iliac arteries located on the umbilical cord. At the placenta, fetal blood picks up O2 and nutrients and eliminates CO2 and waste. Oxygenated blood returns to the placenta via a single umbilical vein, which ascends to the liver. The fetus were divided into two branches. So here is that umbilical vein. Notice what it's colored. Okay, it's colored red because it's containing oxygen and other nutrients. It comes up into the liver. You can see it. Branches, two branches exist. Okay. Let's pick up right where we left off. One branch joins the hepatic portal vein, enters the liver. The other branch, called the ductus venosus, drains into the inferior vena cava. Okay, so here is one. Okay, joins that hepatic portal vein right there. The other one, right there, ductus venosus, drains into the inferior vena cava. Now notice what color we're seeing here in the heart and at here, the inferior vena cava. It's purple. Okay, we call and we denote purple for mixed blood. Okay, meaning it's partially oxygenated, partially deoxygenated. And remember, red is for oxygenated, blue is for deoxygenated. Think back to when you were in elementary school. If you mix red paint, blue paint, it makes purple. And that's what this purple is representing. It's deoxygenated blood, oxygenated blood together, forming what we call mixed blood. Okay. That mixed blood in the inferior vena cava and deoxygenated blood in the superior vena cava, they pass into the right atrium, and instead of passing directly into the right ventricle, some of that blood is able to skip over because of that hole in the heart, what we call the foramen ovale, allows blood to go directly into the left atrium. Okay. Now, some blood does get pumped down into the right ventricle, and then the right ventricle pumps. But remember, there's another lung bypass, what we call the ductus arteriosus, okay, that connects the pulmonary trunk to the aorta. Okay. So most of this blood, however, passes through the ductus arteriosus, the vessel that connects the pulmonary trunk with the aorta. This blood in the aorta is carried to all tissues of the systemic circulation of the fetus. When blood reaches the common iliac arteries, part of the blood flows into the internal iliacs, then into the umbilical arteries, and then back to the placenta for another exchange, which we're showing right here and what we kind of started with. Okay. Now, after birth, we talked about how that hole in the heart closes. This connection between the pulmonary trunk and our aorta, that closes. Okay. We have special names for those things. Okay. And these are two of the 6 major changes that occur after birth when our lungs, kidneys, and digestive organs begin functioning. And those are listed here as 5 and 6. Foramen ovale closes shortly after birth, becoming the fossa ovalis, a small depression in the interatrial septum. And typically this takes place about a year. Ductus arteriosus closes, becomes that ligamentum arteriosum. Other things, the umbilical arteries, they become filled with fibrous cords. We call these medial umbilical ligaments after the umbilical cord becomes tied off. If you think about it, there's no reason to be pumping blood to our belly button because all it is, it's a scar. Okay? There is no connection there to the outside anymore, so there's no reason to deliver blood there. Okay? So these basically become scar tissue called medium umbilical ligaments. There's no blood coming in through the belly button anymore, so that umbilical vein, kind of same thing. It becomes known as the ligamentum teres. Okay, that's number 2. Okay, the ductus venosus collapses, becoming a ligamentum venosum. Okay, remember, there's no blood coming up this way by way of this vein, so that becomes ligamentum venosum. And final thing is that placenta has no function inside a mom, and this is going to be expelled after the babies come out. That's why it's called afterbirth. And it's something that, 10, 15 minutes later, maybe after the baby's born, it comes out. Okay. The doctors do a lot of things with it. They do tests on it in some cultures, and people utilize it for different things. It's not uncommon for people to actually take part of it home and do stuff, make food out of it, which isn't that uncommon in a lot of cultures. Something that my wife and I definitely did not do, but a lot of animals, most animals actually eat it because there's supposed to be a lot of nutrients and stuff. I eat it, I mean, typically it's the mom. Yeah, so if that's something that you want to do, you can do some more research on. Okay, if you guys have any questions, just holler. Otherwise, I'll call it good.